To the IGCSE Biology Hub. In today's class, I'm going to teach you and explain clearly about the topic number 2, which is organization of the organisms. In this class, we are going to cover 2.1.1 to 2.1.5. Let's begin from cell structure. What IGCSE 0610 Biology wants you to know from 2.1.1 to 2.1.5 to describe and compare the structure of a plant cell with an animal cell. Here, limited to certain cell organelles, right from cell wall, cell membrane, nucleus, cytoplasm, chloroplast, ribosomes, mitochondria, and vacuoles. You students should be able to describe the structure of a bacterial cells limited to again cell wall, cell membrane, cytoplasm, ribosomes, circular DNA, and plasmids. We learn how to identify the cell structures such as listed 2.1.1 and 2.1.2 from the diagrams and images of plant cells, animal cells, including the bacterial cells. We learn to describe the cell organelles, functions, and their structures, including plants, animals, and bacterial cells. We learn how to state what cell theory says, stating that new cells are produced by cellular divisions from pre-existing cells, stating that the specialized cells have a specific functions limited to listed cells, ciliated cells, root hair cells, palisade mesophyll cells, neurons, red blood cells, and sperm cells and egg cells, which are reproductive cells or gamete cells. To describe the meaning of certain terms, cells, tissue, organ systems, and organisms as illustrated by examples given in the syllabus. Let's begin. What does the chapter topic name? Organization of organisms means in biology how cells are arranged and forming tissues, organs, and organ systems. Communication and exchange between the cells and biological organelles, which are cellular organelles present in every cells, in order to support the cells and promote continuous life to all living creatures. As you studied before and well understood, cells are microscopic, they are so tiny, and cells are the unit of every living creatures. So we call it as unit of life. Cells are the basic structural and functional units. Cells are the building blocks, or you also define cells are the common denominator of life. Comparing the basic eukaryotic and prokaryotic cell differences. A eukaryotic cell, which is pasted here the left side, has a membrane enclosed DNA, which is the genetic material, deoxyribonucleic acid, which forms a structure called the nucleus, located at the center of the eukaryotic cell. Note the purple DNA enclosed in the pink color nucleus. A typical eukaryotic cells has additional membrane bounded organelles of varying shapes and sizes. So eukaryotic, U means true, karyotic is cell. The true cells means the cell nucleus is surrounded a definite nuclear membrane. In contrast, a prokaryotic cells, which is right side, does not have a membrane bounded DNA and also lacks other membrane bounded organelles as well. Here, this diagram shows prokaryotic cells. You don't see the well-defined nuclear membrane which separates the nucleolus and cytoplasm. This is the diagram. You can see the red color and the circular nucleus define clearly the nucleus and cytoplasm. This slide is all about the plant cells and animal cell, a diagrammatic representation. This is the plant cell and this is the animal cell. Plant cell, you have main distinct salient features are the chloroplast and vacuole. The presence of chloroplast and vacuole distinctly from an animal cells. So both the cells will have many common cellular organelles, but the main notifying cell organelle or the main layer, which is called cell wall, present only in a plant cell, which is not present in an animal cell. This is the main difference, one of the differences. And the second, as I said, the chloroplast present in only plant cell, not in animal cell. Big size vacuoles again present in a plant cell, not in an animal cell. Let's have a look and see one by one the cell organelles. This is the plant cell again. You start a small membrane vesicles, smooth endoplasmic reticulum, ribosomes, and the nucleus, nuclear membrane, nuclear pore, nucleolus, rough endoplasmic reticulum, and this is the Golgi body or Golgi apparatus, Golgi or Golgi vesicles, and the liquid portion is cytoplasm, peroxisomes, mitochondrion or mitochondriae. If you take the chloroplast, you see thylakoid membrane, starch grinds, plasmodesmata, plasma membrane or also called cell membrane, and you have a cell wall. Coming to animal cell, again the nucleus, you have rough and smooth endoplasmic reticulum, plasma membrane, Golgi vesicles, ribosomes, mitochondria, peroxisomes, microtubule, lysosomes, intermediate filaments, and you can see the flagellum, which is sometimes present in some types of animal cells. Again, starting from size, shape, energy storage, cilia, growth, and differentiation, on these topics, let's understand the differences between the plant cells and animal cells. Animal cells range from 10 to 30 micrometers in length, but plant cells range from 10 to 100 micrometers in length. The shape, animal cells come in various sizes and shapes, tend to have a round or irregular shapes, but plant cells are more similar in size and are typically rectangular or cubic shape. How energy stores? Animal cells store energy in the form of the complex carbohydrate glycogen, but plant cells store energy as starch. Cilia are found in animal cells but not usually in plant cells. This is one of the difference. Cilia are microtubules that are aiding in cellular locomotion for cells to move. Growth, animal cells increase in size by increasing in cell numbers. Plant cells mainly increase cell size by becoming larger. They grow by absorbing more water into the central vacuole. In animal cells, only stem cells are capable of converting to other cell types. Most plant cell types are capable of differentiation. This is the bacterial cell wall. This is the bacterial cell. Bacteria are single cell organisms. This means that each bacterium is made up of only one cell. but will perform the entire function. This is very different from humans, whose bodies are made out of trillions of cells. Bacterial cells are much smaller than human cells, can measure from about 1 to 10 micrometer long. Circular DNA is form of closed loop DNA that has no ends. This type of DNA is found in the cytoplasm of most of prokaryotic cells such as bacteria. The plasmid DNA is a classic example of extrachromosomal circular DNA present in a bacterial cell. So bacterial cell consists 70% water and 30% other chemicals. So the other chemicals are highlighted here. So out of 30%, 15% is proteins and 4% is all about small molecules, and 6% RNA, 2% phospholipids, 1% DNA, and 2% polysaccharides. Let's have a look. This is the bacterial cell anatomy. Right from the body is surrounded by a small hair-like appendage that's called pilus. You can see a small projection surrounded by the body, and you can see a capsule, and then the cell wall, plasma membrane, ribosomes, the thread-like, like coil single strand DNA called nucleoid in bacterial cell, extrachromosomal circular DNA, which is called as plasmid, cytoplasm, and a long hair-like appendage is called a flagellum. In plural, it's flagella. This illustration depicts components of a bacterial cell anatomy, including, as you see in the capsule, cell wall, and flagellum. What are plasmids? Plasmids are the extrachromosomal DNA molecules. They are small, circular, and have the ability to replicate autonomously, independently. They are most found in bacteria. Some of eukaryotic cells like yeast and plant cells also contain plasmids. Plasmids are also used to prepare the recombinant DNA with the desired gene to transfer genes from one organisms to another organisms. This is known as, in general, genetic engineering. So plasmids carrying a specific gene are introduced into a bacterial cells, which multiply rapidly, and the required DNA fragment is produced in larger quantities. So let us see the structure of a plasmid. Plasmids, as I said, they are the extrachromosomal but not essential. They are useful but not necessarily present in every organisms of the species. Plasmids are not a part of the genome, and the same plasmid can exist in different species and gets transferred from one another. They have their own origin of replication called ORI, own origin of replication, and they can replicate along with the cell, so that each daughter cell possesses a copy of the plasmid also. Apart from the origin of replication, often it contains genes for antibiotic resistance, for the production of toxins, and other useful genes that may be required for the survival of cells. What is an extrachromosomal DNA? Any DNA that is present outside of the chromosome is called extrachromosomal DNA. It can be either outside or inside a cell nucleus. Example, mitochondrial DNA and plasmids. The structure and function of a cell wall. The cell wall surrounds a plasma membrane and provides the cell tensile strength and protection. Cell wall composition varies depending on the organisms. Plant cell wall are primarily made up of cellulose, that's called the carbohydrate polymer, which is the most abundant macromolecule on earth. Also hemicellulose, pectin, lignin, and soluble protein. Cellulose fibers are long, linear polymers of hundreds of glucose molecules. These fibers aggregate into bundles of about 40, which are called microfibrils. These components are organized into three major layers: primary cell wall, the middle lamellae, and secondary cell wall. Bacterial cell walls are composed of a sugar and amino acid polymer called peptidoglycan. The main components of fungal cell walls are chitin, glucans, and proteins. You can have the diagram to understand the middle lamellae, primary cell wall, and plasma membrane. Pectin, cellulose, microfibrils, hemicellulose, soluble protein. So cell wall is present only in plant cells, not in animal cells. So the functions of cell wall is support, pressure balance, regulate the growth, regulate diffusion, communication, protection, and storage. Let us understand one by one. The cell wall provides the mechanical strength and support the cells. It also controls the direction of cell growth, withstand the turgor. Turgor pressure is the force exerted against the cell wall as the contents of the cell push the plasma membrane against the cell wall. This pressure helps a plant to remain rigid and erect, but can also cause a cell to rupture. Regulating the growth, the cell wall sends signals for the cell to enter the cell cycle in order to divide and grow. Regulating the diffusion, cell wall plays an essential role. It's a porous, allowing some substances, including proteins, to pass into a cell while keeping other substances are out. Communication, cellular communication is essential. So the cell wall involves cell communicate with one another via plasmodesmata. Pores are channels between the plant cell walls that allow the molecules and communicate signals to pass between individual plant cells. Protection, the cell wall provides a barrier to protect against plants, viruses, and other pathogens. Pathogens are the disease-causing microorganisms. It also helps to prevent water loss. Cell wall also stores carbohydrate for use in plants' growth, especially in seeds. Next is the cell membrane. It's one of the cellular cell organelles. Cell membrane is also called plasma membrane. Sometimes it's also called as semi-permeable membrane. It is the structure that keeps the cytoplasm from spilling out of a cell. This membrane is composed of phospholipids, which form a lipid bilayer that separates the contents of cell from the extracellular fluid. This lipid bilayer is, as I said, semi-permeable, meaning that only certain molecules are able to diffuse across this membrane to enter or exit the cells. Extracellular fluids, proteins, lipids, and other molecules may be added to cell's cytoplasm by endocytosis. This membrane surrounds a cell is made up of proteins, lipids. A quick summary: depending on the membrane's location and role in the body, lipids can make up anywhere from 20 to 80% of this membrane, with the remainder being proteins. Cholesterol, which is not found in plant cells, is a type of a lipid that helps stiffen the membrane. The structure and function of a nucleus. This is another main, or you can call it as the brain. Nucleus is a membrane-bounded cellular organelle. This structure contains cells' hereditary information and controls cell growth and reproduction. It is the command center of eukaryotic cells and is usually the most notable cell organelle in both size and function. Nucleus is for control the cell growth and multiplication, regulating gene expression, initiating cellular reproduction, and storing genetic material necessary for all these tasks. The nucleus regulates synthesis of proteins in the cytoplasm through the use of messenger RNA, that's mRNA. The physical characteristics. The shape of nucleus varies from cell to cell, but it's often depicted as spherical. And in an animal cell, it placed in the center mostly, but in plant cells, it's not in the center. The center will be occupied by the big size of vacuoles. So the nuclear envelope and nuclear pores, the nucleus is bound by a double membrane called nuclear envelope. So envelope, membrane, layer are almost same meanings in cell biology. Next, you see the nucleolus, the structure within the nucleus that helps in the synthesis of ribosomes. Nuclear pore, a tiny hole in the nuclear membrane that allows nucleic acids and proteins to move into and out of the nucleus. The nuclear envelope helps to maintain the shape of the nucleus. It's connected to the endoplasmic reticulum, ER, in such a way that the internal chamber of the nuclear envelope is continuous with the lumen or inside of the endoplasmic reticulum. This also allows the transfer of material as well. Yeah, you can see here the diagram of nucleus. As I said, the cell's brain is nucleus. You see the spherical shape, nuclear envelope, double membrane, and this is the nuclear pore, and inside you see the nucleoplasm and thread-like structure called chromatin network, and this is the dense particle in the nucleus or nucleoplasm is called nucleolus. Sometimes in the exams perspective, you students will be asked to label the parts of the nucleus in specific nucleolus and chromatin network and nuclear envelope and nuclear pore. Understanding one by one what does mean chromatin, nucleoplasm and their functions. The nucleus houses of chromosomes containing DNA. The DNA stands for deoxyribonucleic acid, holds the hereditary information and instructions for cell's growth, development and reproduction. When a cell is resting or not dividing, its chromosomes are organized into long entangled structure called chromatin. Nucleoplasm is gelatinous substance within the nuclear envelope, also called karyoplasm, karyonucleus. This semi-aqueous material is similar to cytoplasm in that is composed mainly of water with dissolved salts, enzymes and organic molecules suspended within that. The nucleolus and chromosomes are surrounded by nucleoplasm, which cushions and protects the nuclear contents. Like the nuclear envelope, the nucleoplasm supports the nucleus to hold its shape. It also provides a medium by which materials such as enzymes, nucleotides, DNA, and RNA subunits can be transported throughout the nucleus to its various parts. Nucleolus contained within the nucleus is a dense membraneless structure composed of RNA and protein called nucleolus. The nucleolus contains nuclear organisms, the parts of chromosomes carrying the genes for ribosome synthesis. The nucleus helps to synthesize ribosomes by transcribing and assembling ribosomal RNA subunits. These subunits join together and form ribosomes during the protein synthesis. The functions of cytoplasm. Cytoplasm consists three main components. One is cytosol, organelles and various particles, and granules called cytoplasmic inclusions. So cytoplasm consists of all of the contents outside of the nucleus and enclosed within the cell membrane of a cell. It is a clear in color. It has a gel-like appearance. Cytoplasm is composed mainly water but also contains enzymes, salts, organelles and varying organic molecules. Functions one by one to support and suspend organelles and cellular molecules. Many cellular reactions also occur in the cytoplasm, such as protein synthesis, the first stage of cellular respiration known as glycolysis, mitosis and meiosis will take place in the cytoplasm. The cytoplasm helps to move materials such as hormones around the cell and also dissolves the cellular waste. Cytosol. The cytosol is the semi-fluid component or liquid medium in the cytoplasm. Organelles are the tiny cellular structure that perform a specific functions within a cell. Example of organelles including mitochondria, ribosomes, nucleus, lysosomes, chloroplast, endoplasmic reticulum, Golgi apparatus or Golgi apparatus. Cytoplasmic inclusions, one of the main components in the cytoplasm, are particles that are temporarily suspended in the cytoplasm. Three main types of inclusions. One is secretory inclusions, second is nutritive inclusions, the third one is pigment granules. Examples of secretory inclusions are proteins, enzymes, and acids. Glycogen, glucose storage molecule, and lipids are examples for second type of cytoplasmic inclusions, that is nutritive inclusions. And melanin found in a skin cell is an example of a pigment granule inclusions. So this is all about the structure and function of a cytoplasm. And let us take this image or diagram to see like the distinct difference of nucleus and the gelatinous substance called the cytoplasm. So in the cytoplasm, you can see, as I said, cytoplasmic inclusions and all cellular organelles like mitochondria, ribosomes, endoplasmic reticulum, centrosomes, so on so. Let us move to the next organelle, which is present in only the plant cells. That is called the chloroplast. Structure and function. A chloroplast is a type of plant cell organelle known as a plastids. The chloroplast contain a green pigment called chlorophyll, which absorbs light energy during photosynthesis and converts to chemical energy. Chloroplasts have their own DNA, are responsible for energy production, and reproduce independently from the rest of the cells through the cell division process similar to bacterial binary fission. This organelle, chloroplast, can also be found in other photosynthetic organisms such as algae and cyanobacteria. Chloroplasts are also responsible for producing amino acids and lipid components needed for chloroplast membrane production. The functions and key points of chloroplast. Chloroplasts are chlorophyll containing organelles found in plants, algae, and cyanobacteria. As I said, and photosynthesis occurs in chloroplast. Chlorophyll is a green colored photosynthetic pigment. Within the chloroplast, you can see grana that is ultimately absorbs light for photosynthesis. Come to this diagram. You see a disc-shaped thylakoid and many thylakoids arranged and form granum. So one granum, two second granum, third granum, fourth granum, and in plural it's called grana. The guard cells open and close tiny pores allowing for gas exchange needed for photosynthesis. And photosynthesis occurs in two main stages, the light reaction stage and the dark reaction stage. ATP, which is adenosine triphosphate, and NADPH are produced in the light reaction during the light reaction stage, which occurs within chloroplast, this grana. And in the dark reaction stage or Calvin cycle, ATP and NADPH are produced during the light reaction stage and are used to generate sugar molecules. This stage occurs in plants' stroma. Moving to ribosomes, called the protein factories, protein builders of a cell. Ribosomes are cell organelles that consist of RNA and proteins. RNA stands ribonucleic acid. They are responsible for assembling the proteins of the cells. Depending on the protein production level of a particular cell, the ribosomes may number in the billions and millions. Ribosomes in plants and animal cells are larger than those found in bacteria. There are subunits produced in the nucleus and unite in the cytoplasm during the protein synthesis. You can see the large subunit and small subunit. Free ribosomes are found and suspended in the cytosol, the liquid portion in the cytoplasm, while bound ribosomes are attached to the endoplasmic reticulum. So endoplasmic reticulum with ribosomes are called rough endoplasmic reticulum. If ribosomes are not placed or attached on the surface of endoplasmic reticulum, that is called or known as smooth endoplasmic reticulum. Mitochondria and chloroplasts are capable for producing their own ribosomes. As you can see here, the subunits comprising ribosomes in mitochondria and chloroplast are smaller, 30s to 50s, than the subunits of ribosomes are found throughout the rest of the other cells, 40s and 60s. So you can understand 40s and 60s ribosomes found in all other types of cells, and mitochondria and chloroplast ribosomes are 30s and 30s to 50s. Coming to the powerhouse of the cell, which is called mitochondria. The structure of mitochondria and functions. Mitochondria are membrane-bounded organelles present in the cytoplasm of all eukaryotic cells. That is, the true cells produce adenosine triphosphate, ATP, which is energy, the main energy molecule used by every cell. The mitochondria is a double membrane, as we said. The shape is rod shape or structure found in both plants and animal cells. The size ranges from 0.5 to 1.0 micrometer in diameter. The structure comprises an outer membrane, an inner membrane, and a gel-like material called matrix. The outer membrane and inner membrane are made up of proteins and phospholipid layers separated by intermembrane space. The outer membrane covers the surface of the mitochondria and has a large number of specific proteins known as porins. So the functions regulates the metabolic activity of the cells, promotes the growth of new cells and cells multiplication, helps to detoxifying ammonia in the liver cells, plays an important role in apoptosis. This is called the programmed cell death, responsible for building up certain parts of the blood and various hormones like testosterone and estrogen, helping in maintaining an adequate concentration of calcium ions within the compartments of the cells. It also involved in various cellular activities such as cellular differentiation, cell signalings, cell senescence, and controlling the cell cycle and also cells growth. The most important function of mitochondria is to produce energy through the oxidative phosphorylation reaction. Let's move to the cell organelle which present mostly in the plant cells. Vacuole is a cell organelle found in number of different cell types, and these vacuoles are fluid filled enclosed structures that are separated from cytoplasm by a single membrane. They are found mostly in plant cells and fungi. However, some protista, animal cells, and bacteria also contain vacuoles. These are responsible for a wide variety of important functions in cell, including nutrient storage, detoxification, and waste exportation. So vacuole functions, you see turgor pressure control, growth, storage, molecule degradation, detoxification, protection, and seed germination. Digestive enzymes from the vacuole then degrade the entire cell. Coming to 2.1.5, stating that new cells are produced by division of existing cells. That's what the cell theory states. All living organisms are made out of many cells. If it is unicellular organism, it's made out of only one cell. Cells arise from pre-existing cells, which means the parent cell, and all cells have the basic chemical composition, and cells are the basic units. Hereditary information is passed from the cell. Energy flow occurs within cell. The modern version of the cell theory includes the ideas about the energy flow occurs within the cells, hereditary information, DNA is passed from cell to cell, and all cells have the same basic chemical composition. New cells are produced when a cell called mother cell divides into new cells called daughter cells. When two daughter cells have the same number of chromosomes as the original cell possess is called mitosis, and meiosis is a special type of cell division that halves the number of chromosomes to create egg cells and sperm cells. They are the gamete cells. New cells are created from making new proteins. Cells constantly make new proteins, and many are made before cell divide. Some of the proteins needed to be divided between the two daughter cells so they can continue and to function once the cell division has occurred. Other proteins create the mitotic spindle, which organizes and sorts the chromosomes into daughter cells. Still, other proteins make contractile ring that gradually squeezes this original cell into two cells. Making new organelles, cells also continuously make new organelles, just like they make new proteins, while each daughter cells need to have exactly one copy of each chromosomes. That exact number of other organelles can vary. Copies of endoplasmic reticulum and Golgi apparatus, which together synthesize most of the molecules used by the cell, and mitochondria, which make energy for the cell, and randomly divided between two daughter cells and after chromosomes have been segregated. So students, in this class we have covered IGCSE 0610 biology as per the syllabus guide, topic 2, subtopics 2.1.1 to 2.1.5. Hope this class is very useful, and go through this video again to understand, to get clear informations. And thanks once again for joining the Biology Hub. This is the Fulbright alumni and Cambridge IGCSE biology teacher. Thank you so much.